That's for some technical reason that I don't want to go into detail, but it just looks a little funny. You'll then see after a few seconds that somebody turns on a fluorescent light source. And this fluorescent light source excites a specific set of cells, our dopamine neurons. It excites specifically those because we have made those dopamine neurons express a protein called GFP, green fluorescent protein. It's actually the same protein as in the previous movie where I showed you the one cell. It was just black and white, but it's normally green. It's a protein that comes from thank you very much. Whoever said jellyfish, thank you very much. I was having a little Parkinsonian moment here. So it comes from jellyfish. We can put this into individual cells and thereby visualize those cells. Okay? So again, you'll see the fluorescent light source coming on. You'll see the dopamine cells, and then you will see the bright field come off. You'll then only see the green cells. That's sort of the preparation. Otherwise, it's a little unclear what you can see. Okay, so this is a bright field again. You see it's sort of moving a little funny. Almost like a cat trying to lick its tail here. Now you see those green cells. Those are the dopamine neurons. And unlike us, who have thousands and thousands of dopamine neurons, C. elegans has only 8 of them. You can, if you were to count here, you can actually see there are 8. You know they go in and out of focus, while you cannot see them all at the same time. But basically, what we have here is a very simple dopaminergic system, where we now have the opportunity to ask, how do those dopaminergic neurons develop, right? And again, let me just sort of remind you of the major premise here of our work is. The major premise is that we believe, and we have actually in that specific case very good evidence for the fact, that the genes that control the making of dopamine neurons in C. elegans are the same genes as the genes that do that in more complicated organisms, meaning in ourselves. So how do we find genes in C. elegans? And this is sort of my last thing that I'm going to bring up, and I'll show you a few pictures for that. And what makes C. elegans, or flies, so special in terms of why can we only do this approach in worms and flies and not in humans or mice? It's called forward genetics. So forward genetics sounds a little weird, but forward genetics means it's basically, let me throw a few terms at you and then I'll explain them. You screen for mutants in which something is defective. Right? That's sort of a very global way how to say that. Let's actually start with a fly, because in a fly people have done that quite extensively in a very nice example. People have looked for a long time in flies, who have eyes by the way, for flies that cannot see. So what they simply did is, they threw a very ugly chemical onto those flies, thinking you're basically transforming the fly into a chain smoker, if you want, right? You hit them with a very ugly chemical, very, very hard. Will not, like in humans and with smoking, take 20, 30, 40 years to get a mutation and then get cancer. This is a much more massive dose that it will induce mutations in genes, anywhere in the genome, right? And it will do a very ugly thing. Many flies will die, and it's too bad. That's why we like to work with flies, because we don't feel too bad about it. But we basically then try to find, or not we, people have done it, other people have done that, try to find flies that cannot see anymore. And then the logic leap here is that those flies cannot see anymore because some gene got mutated that is important for them to see. Meaning we then have a mutant fly where people can now study where exactly is the mutation, what gene is mutated in those flies, and by inference we can then argue, well, if you mutate that fly and the fly cannot see it anymore, that means that the normal function of that gene is to enable the fly to see. Right? So this is a very simple example. The more complicated example, but conceptually exactly the same, is this here, where you can take a C. elegans worm and mutate the worm and try to identify mutant animals in which those, actually I'm always pointing to a black screen here, in which those dopamine neurons do not develop anymore. Right? And the idea that again would be, if you have identified those mutants, they will be mutations in genes that are important for those neurons to develop. And I'll show you one mutant where this is actually the case. All right, let's get this up here. Good. All right, so what you see here is a worm, immobilized, doesn't move, in which we have labeled most of the nervous system with actually a different jellyfish protein, a jellyfish protein that fluoresces in red. It's called RFP, red fluorescent protein. This serves sort of just as some sort of a landmark. It allows us to tell us, well, here's the nervous system, right? And it's also a control for us to see that in the mutants that I'll show you, well, they do have a nervous system. That is not a fact that they don't have a nervous system or anything, but it's only the dopamine neurons that are lost. Okay? So again, this is a normal wild-type worm where much of the nervous system is labeled. And just sort of to orient you here, this is the front half of the worm. This blotch here is just a cluster of most of those 302 neurons that I mentioned before. And those thin things here and here, this is essentially their spinal cord, meaning long, thin axonal processes. And this is sort of the overall anatomy of the nervous system here in the worm. Now this here is now a picture of those GFP-marked worms where only the dopamine neurons are labeled with GFP. And those are just 8 of them. Actually, here in the head you can now see 4 different planes of focus. And I should tell you that unfortunately we always have to deal with that stuff. This granular stuff is actually something, quite funny, but it's the way it is. It's E. coli. E. coli just happens to autofluoresce. It's very annoying, but if you shine fluorescent light onto worms, those damn bacteria that they eat here in the gut will just autofluoresce. It's annoying, but because they're so small, you can always tell, this is just the bacteria stuff. So let's overlay those 2 pictures, and you can see here, again, the nervous system and those few dopaminergic neurons. By the way, you shouldn't think that those neurons here are super fat and those are all super small here. It's just because this GFP is so bright and shines so brightly that you see all the sort of the haze around it. Okay.